Hi everyone, thanks for being with us today. As you just heard, I'm Adrian Toutoungi. For those of you who don't know us, Taylor Wessing, we're a law firm working intensively in life science and deep tech. We work with a lot of biotechs in this cluster and around the world, and a lot of investors, including some in the field of longevity. And that takes us on to the topic for today's panel. So longevity is a hot area. We heard about AI being hot, but longevity is equally hot. Probably a year ago now, we heard about Altos Labs coming out of stealth with funding of $3 billion, reputedly. And just 2 weeks ago, I think, the press was covering the news of a riot in Boston at the Convention Center, the International Society for Stem Cell Research. Juan Carlos Belmonte was speaking. He runs Altos' San Diego facility. And the facility had to be cleared because so many people were trying to get into the room. We don't quite have that problem here today. But they had to be cleared because such were the levels of excitement that they just couldn't the capacity was basically overflowing. So we're going to try and drill down to some of those issues today and be informative and provocative in equal measure. Just to make sure we're all on the same page, what do we mean, real basics, by longevity? What we're going to be talking about is extending the so-called health span without age-related diseases. And those are things like cancer, Alzheimer's, heart disease, diabetes, dementia. We're going to be exploring current thinking about the biology of aging and how that might drive and translate into longevity medication. There are other aspects of longevity. You can come at this from all sorts of different angles. And a lot of them are more focused on sort of wellness, lifestyle. For example, there's Brian Johnson, the US tech billionaire that you've probably heard of, is trying to rewind his biological clock, but largely following, as I understand, a sort of wellness routine, actually under the care of a doctor based here in Cambridge, Oliver Zollman. So we're not going to be discussing those type of issues today. We are looking really at the cell level, at the molecular level, at the metabolic level. So I'm going to ask, we've got a really stellar panel of experts today who, as I say, have the brief to be informative and provocative, hopefully in equal measure. And I'm going to ask them to introduce themselves briefly. Delphine.
Thank you very much. I'm happy to be here today. I'm Delphine Larrieu. I'm an assistant professor at the University of Cambridge. And my lab is working on the cellular mechanisms driving ultra-rare premature aging syndromes called progeria. And we are basically studying these mechanisms to also inform us on mechanisms also occurring during physiological aging. I also have an interest in translation, and I'm actually a co-founder of a company called Adjustia Therapeutics, which is based here on this campus.
Brilliant. Becky?
Hi, my name is Becky Canter. I'm a senior director in Lilly Ventures. I have a background in a research background in dementia and CNS diseases. I also have been doing venture investing here in the UK, early stage companies for dementia for the last 5 years. Now at Lilly, I cross all therapeutic areas, so we think about how different chronic diseases really impact people and how we can create medicines to make their lives better.
Great. Panteli?
Good afternoon, everyone. Pantelethiocarus. I'm the Cell and Gene Therapy Strategy Lead for PPD. We're the clinical research group of Thermo Fisher Scientific. Mission is very simple: make the world healthier, cleaner, safer in that particular regard. Started my career in the NHS. I was in the NHS for 10 years, was part of establishing the allogeneic stem cell transplant program at the Royal Free Hospital in London. Took that experience and then moved into biotech and big pharma, developing highly complex therapies from clinical R&D right through to commercialization, pricing, market access and reimbursement. Really glad to be here and be part of this expert panel.
Thanks, Panteli. Daniel?
My name's Daniel. I'm founder of Shift Bioscience, a small company in Cambridge developing therapeutics for cellular rejuvenation. Biochemist by training, quite an eccentric Cambridge geneticist called Aubrey de Grey, gave a TED talk, sort of inspired a generation of scientists to get involved in this field. I sort of followed sort of his path, and that's eventually resulted in Shift Bioscience. Been going about 6 years now, but it's become more exciting in the last 2 years.
Great. Janette?
Hello, I'm Janette Thomas, and I'm the CEO of 5 Alarm Bio. And we're a seed stage company, and we're in our first year of operations. And we're in stealth mode on terms of what our biological mechanism is, but we're doing our research to understand a fundamentally new pathway that we think is involved in ageing, with the aim to look for new chemical entities that would treat diseases of the elderly.
Brilliant, thank you. So that's our panel, a range of expertise, as you can see, from the academic through to some startup companies and investor perspective, as well as the cell and gene therapy perspective. So let's start, I think, with exploring the current understanding of the mechanisms of human aging at a cellular level, molecular level, and metabolic level. So I'm looking at Delphine, because I'm hoping that she can give us a brief summary highlighting the areas where research is currently focusing and which are moving most quickly.
Sure. So perhaps before we start digging into the cellular mechanisms driving aging, maybe we can try to give a definition of aging. So what is aging? And at the cellular level, at least, aging is characterized by a progressive loss of cellular and physiological integrity that basically altogether leads to an inability to cope with stress and therefore to an increased risk of morbidities and mortality. And as Adrian mentioned in his introduction, aging is actually by far the primary risk factor for many different types of diseases. Obviously, neurodegeneration, dementia, but also cardiovascular disorders, diabetes, and of course cancer. And just to give you an idea of the scale of this risk, so if we talk about cancer, for example, lifestyle factors such as alcohol consumption, diet, or smoking can increase the risk of cancer by 20 to 40%. When we talk about aging increases this risk by thousands of percent. Right, so you can see why this is becoming a major issue for our societies, where the life expectancy has dramatically increased in the past century. And so basically the major challenge of aging research and industry is to find ways to extend this health span. So basically this period of life spent free of the chronic disabilities and diseases of aging, right? We are not talking about living forever, we are talking about living healthier. And I don't think anybody will disagree with this concept. So what is required to find ways to extend health span? Basically we need to have a deep understanding of the mechanisms, the cellular mechanisms driving aging. And again, there has been a massive increase in the knowledge of these mechanisms in the past 2 decades or so. And common denominators of aging across species have been proposed and characterized as hallmarks of aging. So they were first proposed about 10 years ago, and at this point there were 9 of them. And they were recently updated actually earlier this year, and there are now 12 of these hallmarks. So what are these hallmarks? They basically encompass processes such as genomic instability, telomere shortening, cellular senescence, but also processes such as chronic inflammation, for example, or deregulated nutrient sensing. And it's really these hallmarks of aging that have led to the development of new anti-aging strategies. So these strategies aim at targeting these hallmarks of aging with the goal of being able to delay the onset of multiple age-related disorders simultaneously. So that's what is called the geroscience hypothesis. So basically, instead of trying to target one specific age-related disorder, like dementia, it's trying to target the biology upstream of these diseases to really delay all of these diseases, right? So the current anti-aging approaches can probably be divided into 2 categories. The first ones being like lifestyle interventions, as you mentioned, with caloric restriction or intermittent fasting and exercise. I would say that currently it's probably the only safe approaches that one can take to enhance health span. But the second one, which is obviously very promising, is a category of pharmaceuticals and genetic approaches. And these basically cover various therapeutics that target specific hallmarks of aging. So for example, cellular senescence with compounds called senolytics and xenomorphics, which will basically get rid of the senescent cells in the body. Other compounds that basically mimic caloric restriction, so that target this deregulated nutrient sensing. And these include compounds like metformin or rapamycin, which are being tested in clinical trial. And other processes such as something called heterochronic parabiosis that you may have heard about, which is basically replacing compounds from old blood from compounds of a young blood. And this probably targets chronic inflammation and stem cell exhaustion. And the last one, I would say, that is worth mentioning, which is a very hot topic in the field currently, is cellular reprogramming or cellular rejuvenation. And I want to hand over to Daniel to tell you more about that.
Yeah, so when I set up Shift, I was originally targeting one of the hallmarks of aging, which is mitochondrial dysfunction, but ended up pivoting the company to work on cellular reprogramming, just to explain why we made that decision. And cellular reprogramming is able to affect all of the hallmarks of aging simultaneously, with a few exceptions. But it's about as close to a single solution that we've had, right, to rejuvenation, not just slowing down aging, but bringing yourself back to a younger biological age. And there's two examples we can look at. One is natural rejuvenation, which is basically having children. So your child is born afresh, even though that you've aged maybe 20, 30 years. So that's quite a mysterious process, but less mysterious recently. And then we have artificial rejuvenation. There's artificial rejuvenation, which is based on something called Yamanaka factors. So 4 protein factors, originally discovered because they can turn adult skin cells into stem cells, but they also rejuvenate those cells in the process. So you can bring cells all the way back to a biological age of 0, but the problem is they're incredibly dangerous. You risk cancer. Nobody wants to be taking a drug that puts you at risk of cancer. So there's this big mystery that we want to unravel, which is how do we get rid of the almost terrible side effects and keep this rejuvenation side. So that's sort of a particular focus of the field at the moment, and Altos Labs is one of the companies sort of setting up to try and find these special genes.
So it sounds like we've got a consensus that understanding the biology is absolutely critically important as we look some way in the future to actually therapeutic effects of the research. Everyone's on board with that?
I think we also kind of draw parallels from other experiences. If we think about oncology drug development, for example, that appreciation of kind of the mechanistic factors, drawing from really great science, translating that for the patient. And from my viewpoint, that's the most important thing for me. How can we engage, how can we influence the patient journey? But it has to all be predicated on, really great science. And the ability, I think, to really, collaborate, to partner, to ensure that we're developing the right tools, not only to kind of engage, really great scientific ecosystem, but I think, the broader spectrum as well. And connecting those dots and making it very tangible and very practical for the patient is, I think, going to be critical to success.
Just to add as well, that there's interventions that you can do yourself, like diet and exercise, etc. But that works best when you're really young. When you're older, your body doesn't respond. So we're interested in sarcopenia, muscle wasting, for example. Older people get frail because they can't put muscle on. So if you can get some sort of drug intervention to enable people to put muscle on themselves when they get older, that could be really powerful. So it's a combination of understanding the biology and having new interventions that we haven't an idea what they are yet.
So we've heard about the mechanisms, or at least some of the mechanisms, and as you say, Delphine, there are 12, did you say now? Yeah. And we've very briefly touched on some of the existing thinking around potential interventions. So you mentioned senolytics, things like dasatinib, which is a tyrosine kinase inhibitor. I think that's a small molecule. And the other small molecules which are also being trialled. Metformin and rapamycin. Yeah. So let's think a bit more then about, so how, having got an understanding of a mechanism, can you just sort of give us some insight as to what form the medications, the interventions are likely to take? Are these going to be small molecules? Are they going to be biologics, like proteins? Are we thinking about cell and gene therapy? It sounds like the ones at the moment that are under most research are actually small molecule.
Yes, it is. I think, gene therapies or genetic approaches, again, when we talk about cellular reprogramming, for example, are still not in the clinics. It will probably take some time, whereas these molecules that I mentioned are already being tested in the clinics, so they are much more advanced. Obviously, we still need some time to understand, whether there might be some side effects and some toxicities, and it's probably going to be tissue dependent because, again, every tissue is going to age differently, and getting rid of senescent cells may be very beneficial for a tissue that's, proliferating a lot, but maybe not for like the brain where neurons are not, not really proliferating. So I think all these kind of things are to be taken into considerations, but yeah, probably other panel members have more.
Are we still too far off clinical deployment for people to be thinking about how these were deployed and administered? So will they be administered systemically or targeted locally? Becky, I'm sure you've come across lots of business plans and people seeking investment from Lilly and in your previous jobs as well.
Yeah, I mean, I think that we see the advent of a lot of, call them new modalities, kind of across lots of different kinds of diseases, and so I think aging is an area where we're going to see that continue to develop, because like you said, it is tissue dependent, it's target dependent, and it's really dependent on how an individual wants to engage with their medicines and how the best way to deliver them safely. But I think as these medicines in other areas start to make their way through the clinic, prove their safety profiles, we're going to see a lot more of that come up in this space as well, especially because there are fewer side effects. You can get things that are more targeted to the right pathways, and so you end up having much more control over what you're able to do with these new modalities. That's the promise of technology. But I think it's really important that it's always the right answer to the right question, rather than having just a blanket, this is new and exciting. We always need to choose the right modality for the right target. And I think that's a big question that we still haven't quite figured out yet. And I think the other piece that I'll mention before I pass it on is, the other piece is the pricing, is that as you start to get into new modalities and you start to get into new technologies, that comes with the cost, be it, the AI costs we were talking about for design or for, trials and all those different pieces. But we have to think about, if you're going to deploy something into aging, which is where you're expecting to deliver that for a long period of time, and as you expand the health span, potentially even longer, you have to be concerned about what that cost is going to look like for that patient and whether people will actually be able to afford those therapeutics.
It's about managing, I think, the patient's expectation.
Yeah.
We saw this in hematology oncology for many years. We want to be able to extend the patient's lifespan. So overall survival has always been a very important clinical parameter. But to the point, I think we also think about it very practically speaking as well. So we need to kind of draw the parallels. And again, in other therapeutic areas, we saw the development of modalities from small molecule right through to highly complex therapies. And we've heard about that in other sessions that have been conducted today during the meeting. So I think we're going to see a continued evolution, but managing the patient's expectation again, has to be part of that process, because the patient really has to be, from my perspective, at the epicenter of everything that we do and everything that we say as we develop these new and exciting and potentially transformational therapies.
And Delphine, you mentioned, so a lot of the research at the moment is at the preclinical stage. People are working in sort of cell cultures, but also animal models.
Yes.
One of the challenges with animal models, I think, is that animals don't age in the same way as humans. So can you talk a bit about that? What animals are people looking at? Obviously, there's been a lot of talking animals who naturally have negligible senescence, so turtles, tortoises, salamanders, those types of things. But what are the difficulties in extrapolating from that into humans?
I mean, I think it's the same as for any other topics, right? Whether you work on cancer or, obviously the pathways can be similar, but sometimes, the end results of compounds might be quite different. So, these hallmarks of aging have been characterized across species. So, it's not just mouse, it's, a range of species, in some cases also in monkeys. And so we know that the pathways involved in cellular aging are very conserved across species. So this is very useful for research, obviously. And I guess depending on the hallmark that you are studying, for example, if if you're looking at telomere shortening and you're looking at a mouse, the mouse has very, very long telomeres compared to humans, much longer than humans, and therefore testing intervention that might work on these specific hallmarks in mice might not be the best. So again, you have to probably pick your model depending on your question. But again, once you actually try it in human, you only have some evidence, which is sometimes, actually for most of these, very strong in these animal models. But you never know how it's going to translate into humans.
I might jump in whilst
might be
remarked. So one of the breakthroughs in this field is being the generation of something called aging clocks. So these are products of artificial intelligence. But what these clocks allow us to do is take a sample from somebody, it could be anybody in the audience, and I'll be able to tell you what your chronological age is within a few years. They're that accurate. And they work across multiple tissues in your body. And now they work across multiple mammalian species. So any tissue in any mammalian species, you can use the same clock. And what this means is you can run an animal experiment with a drug. And you know this clock's going to work just as well in this animal as it is in the human. So there's a direct connection there between what's going on in your animal model and what might happen in the human. So again, we don't fully understand these clocks, but that doesn't mean we should deny their utility. The problem about not knowing what's going on with the clocks is, safety and patient responsibility. So the closer you get to those questions, the more you need to have some sort of black box decoder or interpretability device. But there's nothing stopping you trying to find new therapeutics with a black box. It's just a little bit later that becomes problematic.
Yeah, we're very interested in early sort of cell cultures and model tissues. But people are developing tissues, kidneys and livers and things, but not looking at the aging of those. And we're really interested in that, to work out whether our molecules can intervene in the aging of those specific tissues. And we're working in primary cell culture from patients. And we're looking at specific markers from patient cells. And we see a massive difference in some of these markers between different patients. And you can really see, even in just primary culture, some relatively young people, their cells are really quite elderly compared to older people. And it'd be really interesting to tease all of that out as to what those differences are and why some people's tissues are so much older. It's really interesting what we're finding. So yeah, watch this space with tissue culture and new organelles and things in tissues.
And if I can just add something to that, even if you don't take, someone with an age-related disease, but like a healthy, aged individual, you take 10 of these at the same age, let's say 85 years old, and you look at their cells, it will be very, very heterogeneous. So some of them will look like young cells. Some of them you could definitely tell, okay, this comes from an old person. So there is a massive heterogeneity, which makes the studies even more difficult.
Yes. So you need to QC the cells when you're using them to know kind of predicted age of those cells. Nothing to do with the actual age of the person.
And that's another really interesting parallel because, again, I think in the very early days, on the one hand, we were looking at, storing and cryopreserving umbilical cord blood stem cells for very specific reasons. But now if we look at sort of the cellular sort of therapy landscape with CAR-T, we're always very interested in understanding persistence of these cells. And we talk about T-cell exhaustion. So again, I think there's a lot of parallels that we can draw scientifically and apply to what this means for the patient, both empirically, but also very practically in terms of how we would manage these patients and, again, manage their expectation with regards to overall survival.
And I'm going to just be a bit provocative then and say we keep using the word patient, but aging is something that everybody goes through. So when do you decide that it's something that needs to be treated? And I think we have more questions than answers, so I'll just preface that. But how do you decide when it's something that it needs to be treated, that there should be a drug, there should be a therapeutic for this, versus this is something that is natural, especially when you see the diversity of aging, and I'd say there's chronological age and biological age. When you look at the biological age across different tissues, does it match? Does it not match? What is the trigger that makes you think now we need to intervene? And what is healthy aging then? If we try and reverse aging, what is healthy aging? And what actually pushes us forward in maturation? I think those are questions that we need to ask ourselves as we think about what the biological mechanisms are is like, well, then if we define aging in this way, how do we, like, what is aging as a pathology versus aging as a human phenomenon and a life phenomenon?
Yeah. And what would, thinking ahead to when these interventions do enter the clinic, what would the endpoints for the trials be? Daniel, you talked about a biological, the clocks you were talking about. Could they be a surrogate endpoint? Would they be accepted by the regulators? Could you demonstrate an effect on the data coming from that AI-generated model? Because obviously the actual clinical endpoints would result in very long trials and very expensive trials. So what's the thinking on that, or have people not really thought that far?
Yeah, so there's an ongoing debate. Do we try and sort of reinvent the regulatory framework to recognise things that weren't previously disease as disease? Try and convince regulators that getting old the way we historically have got old is a tragedy, right? Nobody should be subject to it. So that's one approach, and there's lobbying efforts. There's all sorts of things going on. But then you also have a different approach, which is certainly a cellular reprogramming. Not only does this biology seem to reset the hallmarks, it goes beyond the hallmarks to disease phenotypes. There's different names for this, like synthetic rescue, or rescuing disease back to health. But you can basically tread a traditional regulatory path just by targeting a certain disease. So certainly for the Yamanaka factors, which are sort of the proof of concept example, there's data for the brain, the cartilage, the liver, and the muscle that you can actually affect disease phenotypes in sort of different models, which is very promising. It means you could get something to market and perhaps through secondary approvals start spreading around the rest of the body, and then maybe there's some off-label access to people that aren't characterised as pathological, but they want access to those drugs. So I don't know exactly which way it's going to go, but there's multiple routes. Yeah.
So let's talk a little bit about investors and sources of capital, in case anyone in the audience is enthused enough by this to decide that this is an area they want to go into and set up a biotech. So who is investing in this field? Is this sort of the classic biotech specialist VCs, institutional VCs, or other sources of capital coming to the fore? Daniel, I know you've got some views on this because you've been fundraising.
Okay, I'll take this one and then I'll sort of let somebody else talk. So initially, investors, there was a personal dimension or an enthusiasm dimension. So I just take the example of Shift. Jonathan Milner, who's sort of an angel in Cambridge, well recognised, sort of the reason he invested in us, where otherwise he wouldn't, was that he sort of felt he could have a big impact, like a disproportionate impact. Very high risk, but if you could do this, it has all sorts of repercussions. That's one example. You've also got other interests, like there are people that can't take it with them, so maybe they're going to invest a bit because then they can enjoy their retirement a bit more because it's longer. There's all sorts of motivations. Whether that's right or wrong, it's sort of that's a secondary discussion. But yeah, you've got this big enthusiast element, and then there's the economic side of it, which is if you can actually find this biology and target multiple diseases, it's an incredibly efficient type of medicine. It's almost like more of a universal medicine versus a personalised medicine. The great thing about universal medicine is that it applies to all of us, and therefore it doesn't need to be priced at a huge level to recoup R&D because everybody's the market. So ironically, you can actually price it much lower to achieve a better return.
That's interesting, and certainly the enthusiast aspect that you mentioned is something that resonates with me. We've seen a number of, from our fintech practice, a number of crypto dudes who are quite into longevity in a way that they wouldn't think of other areas of life science. But Becky, what have you seen? Because obviously, both at Lilly and beforehand, would you invest? Have you invested? Or is this not really an investable proposition yet for a VC?
Yeah, I think that there are actually a lot of specialist VCs out there who are investing in longevity and aging. There's a number of funds who are focused on those questions, and I think there's both kind of the promise of these big drugs that can help to reverse or slow aging, and then also the insights that are generated by that kind of science. And so can you then actually create drugs that actually target things that are very acute for people right now, be it the chronic kidney disease, heart failure, dementia. Those are things that we have an understanding of the mechanisms. We're starting to push drugs into the clinic. And so where we see the kind of overlap between the science and maybe more near term, I say near term being 5 to 10 years, but targetable disease phenotype or disease that we see as a more classic approach to targeting age-related diseases. I think when we start to look at that, you can see where the VC interest might be, because there is interest then from the pharmas of the world. You know, how do we bring drugs to help people sooner rather than later? You know, the horizon needs to be both long-term and also short-term, because people continue to age, they can continue to have symptoms, they continue to, you know, their lives are getting worse because we're not delivering drugs. So we have to be quite speedy about it. We can't wait for that like one medicine that's going to treat everything. We really need to kind of go after it, both short-term and long-term. I think the VCs have that horizon, so that's quite nice.
I think it's being able to kind of continue to kind of challenge that scientific boundary almost. And I agree with you. We've kind of seen this in other areas where we move from ultra rare to rare indications into more chronic conditions based upon the science of them and the sophistication of the modality. So I agree. I think it's about timing in terms of intervention. But if we have something that has a very strong benefit risk and is going to be cost effective because of the broad application, I think that's a really great value proposition for patients and for other stakeholders within the ecosystem.
Yeah, actually thinking about that, there was one other thing that I was just thinking about is that, you know, when we think about how you invest and how you move medicines forward, I mean, it's all always going to be based on the data and the science. So it really goes back to the heart of this panel, which is you need to understand the biology, and you need to have good data, and you need to have strong reproducible data that underlies the science. And as we start to develop that in this field, I think that is where you start to see the investment come through, is, you know, being able to show that this is a true scientific endeavor with key endpoints and trials and preclinical studies. And that's where you see investment follow, is where the science is breaking. So it really does come down to that data and the strength of that work that's being done.
Janette, can I just take Janette first?
We just raised a seed round, small amount of seed money. Thank goodness we've got some fantastic angels in the UK, just so brilliant. And they're prepared to, they like the idea that it's innovative and new science, and they're prepared to back it even though it's risky because we don't know where it's going to go. And I just think it's fantastic in this country for that. But the next round, I think I'm hearing from people I'm talking to, it's the same as it always is in new drug development. You need to know your target, you need to know the science and the biology, single target, just normal drug development, really. So it's the same model for VCs and bigger investors as it ever was. You've got to have that right business model, that drug development program. You're going to go for the one indication at the end of it that the regulators have given you the rules about how to get through to market through the clinical trials. So that bit's not innovative. You start with the innovative science, but the rest of it is the same pathway.
But does that, coming back to what Daniel was saying, does that mean that at this point you're having to identify an existing well-recognised disease of ageing, and that's what your target label is? You're not going to go for ageing on your label.
Yeah, there's no ageing in that you can, at the moment, that you could, the regulators will approve. There's no ageing disease, although there is a study ongoing looking at that. But we need to, and I think they're looking at this, regulators are trying to get more creative. I mean, I think combinations are going to be important to treat diseases of the elderly, because, don't have one issue with one target when we get older. We need probably combinations would be an easy way in, but yeah, more ways to get through the regulatory pathway, I think.
So we could take the panel in all sorts of different directions. We could talk about the ethics of all these interventions that we're talking about, which we haven't touched on at all. We could talk a bit more about the regulatory and reimbursement aspects, but we've only got 2 minutes, I think. So, and we did want to try and make this as interactive as possible if we weren't trying to make up time from this morning. So have we got any questions for the panel? Gentleman here at the front.
So, yes, this is a slightly facetious question.
To the microphone, yeah. Oh, that's right, Andy. Oh, you've got 2 now. Thank you.
So, yes, this is a slightly facetious question, and it's the balance between prevention and treatment. You had a brief discussion about, are these people patients or not? And I just wondered, you've talked very much about drug intervention, but how do you streamline that with other forms of prevention? You could title that, how do you eat burgers and pizzas every day and live to be 100?
I'm going to be, I could take that, because I can be a bit, so, Lilly has drugs that we use kind of, along those lines, how do you eat, kind of what you need and what you want, and then on the other side, you look quite good. Physiologically look quite good, I should say. And I think, there is that question between the prevention and the therapeutic, but at the same time, where do you start, right? What is prevention when you're talking about aging? Because you start aging the second you're born. If you think about aging from a biological clock perspective, you start to gain those methylation marks. You start to see all those different pieces on your DNA the second your cells start dividing. And so where do you start prevention? I think that is the question. But I do think that there is, when we think about drugs and deploying them after the clinical trials, we include lifestyle management as a huge component of that, be it through behavioral, perspectives, through digital tools. And so there is, there's the therapeutic piece of it, but wherever there is a therapeutic intervention, there's also that huge piece of the care and the clinical care that patients and people should be receiving from their physicians. And there's a question of what kind of physician and where do you get that? Maybe you have some thoughts.
I mean, just kind of drawing from my own personal experience, I think when we kind of think about the clinical development paradigm, if we take about, again, if we look at cancer, for example, we're really focusing on these patients, these poor patients with very advanced, very sort of metastatic disease. But I think every sponsor wants to be able to move their asset into an earlier line of therapy and give the patient the best chance of success. And that relates, again, to overall survival. So I think it's going to need to be a very pragmatic approach, but I think engagement with the regulators is going to be key as we continue to really understand and ascertain, the power of the science behind this really fascinating area of clinical development.
Great. Well, I'm afraid that was the only question we had time for. I did also ask the panellists to prepare a one sentence of wisdom that they could leave you with, but we don't have time for those either.
Well,
So
it's very quick.
no. People can
sold it now.
people can ask them over coffee.
Oh, okay.
So that's an incentive to come and say hello. So thank you for your attention. Thank you very much to the panel, and a big round of applause, I think.